Many thanks indeed, Deborah, and also for Mary for inviting me. Really a great pleasure to be here this morning. What I'm going to do is really take up some of the themes that we've heard from Mick and then David and then Julie and talk about light, sleep, its regulation, and also how the sleep systems are interacting with the light regulation of the clockwork. And the work I'm going to present is the product of an interaction between 4 groups within the Sleep and Circadian Neuroscience Institute. We've got Arti Yaganath, Sri Vasudevan, Stuart Pearson, and myself. And what I thought I'd do in the next 35 minutes or so is have a brief introduction of just 1 or 2 slides here, but actually present the whole talk in one summary slide, because it's kind of complicated. And then with that framework, we can then dissect it and talk about the light regulation of PER1 and PER2, these key targets of light regulation of the molecular clockwork, via 2 pathways, Cre activation and AP1. And the AP1 stuff is unpublished. Then move on to adenosine regulation of PER1 and PER2, and essentially how adenosine sort of hijacks part of the light entrainment pathway. And because of the role of adenosine in sleep-wake timing, talk about the implications of sleep and circadian interactions, and then really ask the question, can we use some of this mechanistic understanding to develop new therapeutics to provide, in a sense, a pharmacological replacement of light for those who are profoundly blind? Okay, so let's kick off with this introduction and overview. And as I'm sure everybody here will be aware, that, of course, if you expose a mouse or a human to a light-dark cycle and they have no rods and cones, then you can still achieve entrainment. Now, it doesn't say that the rods and cones play no role, but they are not the primary way in which the retina detects the light-dark cycle for the regulation of internal time and the entrainment pathway. That's dependent upon a relatively small number of photosensitive retinal ganglion cells, which utilize the photopigment melanopsin. But the topic of today really is going to be, how do those PRGCs interact with the molecular clockwork in the master clock within the suprachiasmatic nuclei? And this is essentially the whole talk. So what we'll talk about is light activation, the release of glutamate and PACAP from those PRGCs, how that increases calcium and cyclic AMP intracellularly in SCN neurons, and how that light signal essentially interacts with 2 regulatory elements on PER1 and PER2, an AP1 and a Cre binding domain. Here's the EBOX domain that Mick was talking about that binds clock and BMAL. So the first element is the activation of CREB and CRTC1, whose proteins bind Cre, and you get the first sort of wave of PER production. Then there's this second limb. Now, CFOS gene has a Cre element, so it will also be activated by CREB and CRTC1 to produce its protein FOS, but FOS can't bind to AP1 alone. It requires a binding partner in the form of Jun, but Jun has to be phosphorylated. And that phosphorylation comes about by the activation of ERK, which then combined phosphorylated Jun can then bind FOS, and then we get activation of AP1 and a second wave of PER production. So that's, in a sense, the light activation pathway, and then we'll talk about how that is turned off via SIC1, and that raises some interesting issues in terms of internal desynchronization, and actually a theme that David kicked off with. And then the last part of the talk will be on the fact that there are 2 types of adenosine receptor on SCN neurons, an A1, and there's about 5 times as many A1 receptors as there are A2A. The A1s, via inhibitory GI pathway, reduce the levels of calcium and cyclic AMP upon binding their ligand adenosine, whereas the A2A actually excite the production of calcium and cyclic AMP. So these 2 are in opposition. Collectively, this pathway will then change the molecular clockwork, and depending upon when you see light, or indeed when you see adenosine, as we'll discuss, you can either advance or delay the clockwork. And we'll see this scheme as we go through the talk, and hopefully I'll layer the information. Okay, so the first point is that glutamate and PACAP give rise to an increase in calcium and cyclic AMP within SCN neurons. How does that occur? Well, there's three basic pathways that have been elucidated. We've known for a long time that light activation gives rise to the release of glutamate, and some wonderful work from Jens Hannibal in Denmark on the role of PACAP. Glutamate is detected and elicits a response via two mechanisms. There's the long understood NMDA receptor pathway, a voltage-gated, a ligand-gated calcium channel. It opens up upon binding and calcium will flood in from the outside. And this is something, in fact, I worked on for years ago with Chris Colwell and Mike Menaker. The second glutamate activation is via glutamate receptor 1 pathway and via a GQ and phospholipase C pathway, you get the release of calcium from internal stores. So there's two ways in which you can regulate calcium. And then, as I said, PACAP via its receptor and a GS pathway, which interacts with adenylate cyclase, gives rise to increased levels of cyclic AMP. So in a sense, the light activation and the primary signal is calcium, an increase in intracellular calcium and cyclic AMP. Okay, let's now see how that pathway interacts with the PER1 and PER2 regulation. And let's kick off with Cre activation. And again, I'm going to be sort of illustrating this using this pathway. We're going to be talking about calcium, CREB-CRTC1, CREB-CRTC1, and the first wave of PER production. Much of this is what I'm going to show you has been published in 2013 and led by Arti Jagannath and Stuart Pearson, and I shall show you some unpublished data that supports these early findings. Okay, so here's the schematic, and then I'll show you the data. So as I said, you've got the release of glutamate, PACAP, intracellular calcium, and cyclic AMP. Now, what we knew before is that CREB is phosphorylated via pKa as a result of this changed intracellular signal. But the key thing is that CREB cannot act alone. It cannot bind to the Cre domain alone. It requires a partner. And what we found is that in addition to phosphorylating CREB, CRTC1 is then dephosphorylated by calcineurin. And in that state, in the dephosphorylated state, it can move into the nucleus and then bind with phosphorylated CREB. So these two can interact when CRTC1 has lost its phosphate group. It can then bind to the Cre domain, and then you get a wave of PER production. And you see the rise of PER, and I've illustrated PER1 and PER2 here. We'll come back to this differential activation later on. But the key point about this slide is that both PER1 and PER2 after activation are then turned off. So what's the mechanism that essentially makes this system non-responsive to light? What's the effects of, what's the mechanism whereby there's a break on the effects of light on the clock? And that break is generated by SIC1. SIC1 is a gene that also has a Cre domain. So like PER1 and PER2, it will bind phosphorylated CREB and CRTC1 in the following way. That leads to transcription and translation. SIC1 is a kinase, and what it does is rephosphorylate CRTC1. And of course, in the phosphorylated state, CRTC1 can no longer bind to phosphorylated CREB, so it falls off, which means you lose the transcriptional drive, which means you effectively turn off this whole system. And of course, SIC1 is also being regulated by a Cre domain, so it turns off its own transcriptional regulation. If you remove light, then you've lost your glutamate PACAP signal, no more calcium and cyclic AMP, and the whole system goes back to the resting state. Okay, so let's now look for the evidence for those statements. And just to remind you, what I'm talking about is that light hits the molecular clockwork, you get the rise of PER1 and PER2, SIC1 kicks in, it phosphorylates CRTC1, and then essentially turns off the transcriptional drive on these two genes. Okay, and in a sense, what this is the molecular basis of jet lag, and we'll look at some of the jet lag protocols fairly shortly. Okay, so this is the in vitro evidence. Here's our little schematic. The first is that what we would expect is that after activation, that CRTC1 will be able to move into the nucleus, where it will then bind with phosphorylated Krebs. These are some lovely pictures from Steve Hughes, which I couldn't resist showing you. This is pre-activation. CRTC1 is distributed evenly throughout this cell. After activation, it moves beautifully into the nucleus, where it has its effects, of course. So yes, there's support from that side. We'd also predict, of course, that SIK1 will be upregulated upon activation of these cells. And indeed, you see a nice activation of SIK1, which very broadly parallels the activation of PER1 and PER2. The second thing is that if we knock down SIK1, as we've done here, what we see is that we see high levels of PER1, in this case, induction, both PER1 and PER2. And so if you knock SIK1 down, it's essentially you've taken away a repression of the system and you get upregulation. So again, the fact that SIK1 is acting to disinhibit, as it were, or rather inhibit PER1 and PER2 production is supported by these data. And then finally, you would predict that SIK1 is actually required for the phosphorylation of CRTC. And these data show that at 0 time here, there's fairly low levels of SIK1, and you get this level of phosphorylation of CRTC. 120 minutes, you get high levels of SIK1, and we see a significant rise in the phosphorylation of CRTC. So all the in vitro data fits very nicely with the model. But the key thing, I think, is the in vivo evidence. And so what Artie did was to use a sort of a knockdown approach, whereby using double-stranded RNA, RNAi, to locally inhibit the production of SIK1 around the suprachiasmatic nuclei. And she achieved pretty effective knockdown of SIK1. So we had a mouse with low levels to vanishingly small levels of SIK1 in the SCN. And this allowed us to do the following experiment. And so here we see the jet lag protocol. Here's the control, here's the light-dark cycle. And in this case, the light-dark cycle was advanced by 6 hours. And so you see entrainment, the light-dark cycle was shifted here, and you see a re-entrainment after around about 6 days. And this, of course, is the cartoon, and I will show you the data in a moment. The same protocol for the SIK1 knockdown. The light-dark cycle was advanced by 6 hours. And what was so remarkable, and I have to say very pleasing, is that what you saw is a very rapid re-entrainment. Took about a day to adjust to the new and shifted light-dark cycle. So that's the cartoon. Here's some actograms, seeing the 6-day transition versus the very rapid readjustment. Okay, so that data has been published, but some unpublished data using a SIK1 mutant mouse, very generously donated by Philip Cohen and Chris Clark at the University of Dundee. So here we have a mouse that doesn't have SIK1 at all. And so the key thing was, can we replicate those knockdown findings that we found in the earlier report? So again, we replicated the jet lag protocol by advancing the light-dark cycle by 6 hours. So we see the 6-hour advance here. And again, about 6 days for this mouse to readjust. This is the control. The mutant mouse, to our great pleasure, realigned itself just like the RNAi knockdown mouse and took about 1 day to adjust. So that's a replication of the original findings. But because this mouse, had no SIK1, we could do longer-term experiments. And so the next set of experiments was to use a light-dark cycle of different irradiance. So here's a light-dark cycle where the light portion of the light-dark cycle was 100 lux. We then advanced that light-dark cycle, but we then lowered the irradiance of the light portion. And in the following sort of manner, we advanced sequentially, but with increasingly low levels of light. And basically tried to define how our SIK1 versus control animal would lock on, and whether it indeed could lock on to this rapidly a changing light-dark cycle with diminishing intensities. These are the control animals, and you see entrained here. It takes, again, this slide to this relatively slow adjustment. It adjusts, then another shift, and you're basically getting into this area here where there's very little evidence that the mouse is actually locking on to the shifted dim light-dark cycle. By contrast, the SIC1 knockout showed an extraordinary and ready realignment to the new light-dark cycle. In fact, not only could it rapidly re-entrain, but also we saw enhanced sensitivity. So at sort of 1 lux, the mouse was perfectly capable of entraining to this light-dark cycle. So SIC1 mutants do show enhanced re-entrainment to a jet lag protocol over 100 to 1 lux range. So that's sort of where we are with SIC1, but it raises, I think, some interesting questions that we may want to address during the question time. So why bother to have a break on light at all? Why not just simply shift the light-dark cycle to, shift the circadian system to the new light-dark cycle immediately? And what's the downside of removing this break of SIC1? And of course, where we may be sort of wondering, I think, is during the discussion, is internal desynchrony, perhaps. What's going on in the central versus the peripheral entrainment mechanisms? Are we seeing a similar sort of buffering of entrainment, of realignment, using a SIC1 type a negative feedback loop? We don't know yet. And indeed, it isn't on here, but what's happening to the sleep-wake cycle as we advance the rest activity cycle or the circadian cycle through those 6-hour shifts? And we're working with Vlad Varzovsky to address that question. And Lewis Taylor is getting answers to all of those questions, but we don't have them yet. Okay, so we've talked about Cre-activation and the turning off of the system by SIC1. Let's now go to this second pathway, AP1. So this is what we've seen, and now what I want to talk about is this activation of FOS, its binding to phosphorylated Jun, and activation of AP1, and this second wave of transcription. So the AP1 domain and its activation by FOS has sort of kind of been around for a long time. I mean, early studies by Benji Rusak and Joe Takahashi, led by John Kornhauser, in the 1990s, show that after you hit the after you hit a mouse or expose a mouse to light, you get FOS induction within the SCN, and it's been used as a very powerful marker. We've known also for a long time that ERK activation is required for entrainment at some level, and Martin Ralph's studies show that if you knock down FOS in a FOS-less mouse, you would attenuate by half the size of the phase shift that you get in mice. But for reasons what I'm not completely sure about, AP1 regulation of PER1 and PER2 fell out of fashion. And certainly a lot of the early sort of the FOS results were interpreted as, well, it's simply a marker of light activation. It plays no fundamental role. We became interested in this whole AP1 activation a few years ago as a result of working, and Artie went to work with Uli Schibler using this star-prom technique. And I won't bore you with the details, but essentially it's a phenomenal technique that can essentially identify transcription factors controlling various pathways. And so what we had access to within previous experiments was essentially the light-activated genes within the SCN. And I won't bore you with the details, but in a sense, these are genes that have been activated within the SCN. And a significant number of those genes showed activation of the AP1 domain. So here's the star-prom result, here's an AP1 domain, and just for the sake of completeness, here's PER2, and you see this AP1 domain, it's a very short sequence, is highly conserved in humans, mouse, dog, elephant, and armadillo. I'm not quite sure why we had armadillo there, but anyway. And so AP1 was clearly doing something, and doing something important. And again, I'll go through the cartoon. I don't have a huge amount of time, so I'm not going to provide very much data for this part of the talk, but this is all now submitted for publication, so you'll be able to get the details later on. So we're sitting in the SCN. There's no light exposure, and we've got these two genes there. There's the CFOS gene and the CJUN gene. The CFOS, as I said before, has CRE domains. The JUN has an AP1 domain. So with no light activation, not a lot's happening, of course, except for one very important thing, is that CREB, in the unphosphorylated state, sits on the AP1 domain and actually inhibits transcription. So it's sitting there and inhibiting transcription. So let's now move to the light activation domain. So here's calcium, cyclic AMP, our critical signal. We've got CFOS. And so just as we've seen before, pKa phosphorylates CREB. CREB then binds dephosphorylated CRTC1. And so we have a first wave of CFOS transcription and translation. So early on, you get a big bolus of FOS induction in response to light via activation of CRE. There's a slower activation of JUN. So what happens, first of all, is that pKa phosphorylates CREB, and in the phosphorylated state, it moves off of the AP1 and essentially stops inhibiting it. So we get then low levels of CJUN. So, okay, great, we've got the two binding partners here, but CJUN can't effectively bind FOS until it's phosphorylated. And so this calcium pathway here activates ERK. ERK then adds a phosphate group to JUN, and in the phosphorylated state, we can get binding to the AP1 and increased levels of JUN production. Of course, with that increased level of JUN production, we then get a reinforcement, and we can get even fur. So you see that sort of rolling wave of JUN production, which enables it to then bind to phosphorylated JUN production, which enables it to bind to FOS, and then the two can work together to activate AP1 on those PER1 and PER2 genes. So let's now look at what's going on in total. So here's our PER1 and PER2. We've talked about the CRE. We're going to now see how FOS and JUN interact with that AP1 to give that second wave of PER1 and PER2 production. So just for the sake of completeness, here we have our phosphorylated CREB binding dephosphorylated CRTC1, binding to CRE and producing a certain level of PER production. Our FOS production is there, but it can't actually activate AP1 until it combined with phosphorylated JUN. And so that takes a while, and we again get a second wave of PER production. So that's how the two regulatory domains of CRE and AP1 are generating PER production. But one thing that struck us as being rather intriguing is that could the differential activation of CRE and AP1 explain the differences in PER1 and PER2 levels of expression? What we've known again for some time, and this happens to be a picture from Mary Harrington's group, is that PER is induced relatively rapidly and then declines, whereas there's a slower rise and decline of PER1. And that's been sort of turned into a cartoon in this figure here. And so what Artie then went on to do is look at the CRE and AP1 domains in PER1 and PER2. And what was very intriguing is that if you look at the CRE and AP1 domains, what's very clear is that the CRE regulatory elements in PER1 are much more accessible. And in fact, whilst there could well be AP1 regulatory domains, I can't say AP1 regulatory domains in PER1, they're not absolutely obvious. And it's not absolutely clear that PER2 has a CRE domain. So what sort of seems to be happening is that PER1 production is being dominated by CRE regulation and PER2 via AP1. And so that would kind of explain that differential activation, that rapid turn on via CRE is producing that sharp rise and then turn off of PER1, and then via AP1 a slower rise and turn off of PER2. The biological significance of that differential activation of PER1 and PER2, again, is something we might want to discuss. Okay, so that's the light activation of the pathway. Let's now go on to unpublished data on adenosine regulation. And let me stress that this has also been driven forward by Arti and Shri. And again, what I want to add to our picture here is the roles of the A2A and the A1 adenosine receptors. So, as many of you will be aware, the two-process model of sleep regulation is sort of conceptualized in this cartoon here. So from the moment we wake in the morning, there's an increase in sleep pressure, and part of that sleep pressure comes about as a result of the role of adenosine. So adenosine, sleep pressure builds and builds and builds throughout the day, and then during sleep pressure and levels of adenosine diminish. And evidence for a role for adenosine in the generation of this homeostatic drive for sleep comes from a very nice, I recommend you see this review here published last year. But in brief, adenosine introduced in the brain will promote sleep. Adenosine increases within the brain during wake. Adenosine dissipates in the brain during sleep. And adenosine receptor antagonists, such as caffeine, will promote a greater sense of wakefulness. Now this homeostatic drive for sleep is opposed by, in a sense, the circadian drive for wakefulness. So in the morning, we see relatively low circadian drive for wakefulness, and it doesn't have to sort of work very hard because the homeostatic drive for sleep is fairly low. But what happens is that the circadian drive for wakefulness increases throughout the day as the homeostatic drive for sleep increases. And of course, in the late afternoon, early evening, the homeostatic drive for sleep is extremely high, but it's opposed by the circadian drive for wakefulness. As we fall into sleep, the circadian drive for wakefulness diminishes. During sleep, it's very low, but then as we approach wake, it increases. And of course, the sleep window is defined by these interactions between the homeostatic drive and the circadian drive. The key point I want to flag up here is the role of adenosine. As I said, there's an A1 and an A2A receptor within the SCN. And the key point is that there's 5 times more, and maybe more, A1 receptors compared to the A2A receptors. And the A1, remember, are the inhibitory input onto that light regulation pathway. So what we see, of course, is with wake, an increased level of adenosine. But the net effect is that the adenosine will differentially activate the A1 pathway and diminish the levels of calcium and cyclic AMP. So you get lowered levels of calcium and cyclic AMP as a result of wake and high levels of adenosine, which then acts to effectively not turn off, but certainly inhibit that whole light-induced pathway. Okay, so what I'm saying is wake increases adenosine, the A1 receptor, you get reduced cyclic AMP calcium, reduced PER1 and PER2, smaller responses to light. Let's test that hypothesis with some data. So in this experiment, very straightforward, we have a 6-hour sleep deprivation during the second half of the light phase. And what we'd predict is that we would increase adenosine, and therefore we'd see smaller responses to light. We'd reduce PER, and we'd see smaller responses to light. And certainly you get lower levels of PER1 and PER2 after sleep restriction. What happens if, so sleep deprivation suppresses PER1 and PER2, entirely consistent with the model I've shown you. What happens if we do this experiment? So we've sleep deprived here, we see, in this case, lower levels of PER2, but we're going to use an antagonist, which will then block the effects of adenosine on this pathway. And the result is a huge increase in levels of PER2 when you've used an antagonist. Essentially, you've blocked the effects of adenosine on this pathway. So, adenosine receptor A1 antagonists reverse the effects of sleep deprivation. Here we see some behavior. So in this case, we've got a vehicle, and we're then going to add an antagonist. And again, what we said is it will block or reduce the effects of adenosine and potentially increase light sensitivity. So we'd predict that an antagonist would produce larger responses to light in terms of behavior. And that's what you see. You see this beautiful reentrainment to, again, this shifting jet lag protocol. So adenosine receptor antagonists enhance reentrainment. Oh, okay. And so again, we would expect that an agonist, which will mimic the effects of adenosine on this pathway. Here we see the size of a phase shift in response to light. We're then going to add an agonist, which will mimic the effects of adenosine, and we can actually pretty much abolish the effects of light on the clockwork with an adenosine agonist. So agonists attenuate the phase shifting responses of light. So what we got is lovely evidence for the image I showed you earlier. Wake drives up levels of adenosine, and via this pathway essentially turns off the whole light-induced effects on the molecular clockwork. Okay, let's look at the implications of this for sleep and circadian interaction. So the key point is that sleep-wake history, encoded by levels of adenosine, acting via the A1 and A2A receptors, modulate the responses of the molecular clockwork to light. Implications for the PRC. Just for those of you, I'm sure you all are familiar with these sorts of images, but as you know, different light pulses at different times, either during the middle of the subjective day, will produce relatively small effects of no phase shifts. The early subjective night delays, further into the subjective night, bigger delays, but towards dawn, you get phase advances. Now, I guess you'll already be with me on this one. What we're saying is that with increased levels of adenosine during wake and diminished levels of adenosine after sleep, it will mean that one's sleep-wake status will have different effects upon the effects of light on the clockwork. So there'll be, at the end of the wake phase, with high levels of adenosine, a maximal suppression of PER, whereas after sleep, there will be minimal suppression of PER. So what do we see in a nocturnal species, such as a mouse? Well, it's awake at night. It then sort of finishes its activity, high levels of adenosine. It then will have high levels of adenosine at dawn. So we'll see small phase shifts at dawn, whereas, of course, after the sleep phase during the day, there will be low levels of adenosine and therefore large phase shifts at dusk. And indeed, what's striking about many nocturnal phase response curves is that there are large delays at dusk and small advances at dawn. Whereas, of course, in a diurnal species such as ourselves, you get the reverse. So at the end of the wake phase, we have high levels of adenosine at dusk, and of course after the sleep phase, low levels of adenosine and big phase shifts at dawn. And in fact, that is a characteristic of many diurnal phase response curves. Now, I'm not saying that the complete phase response curve is being driven by this interaction of sleep-wake, but the fine-tuning of the phase response curve is clearly being influenced by the sleep history of the organism. Okay, let's just finish up on the last few slides with whether we can use these sorts of information to develop new therapeutics. And following on from our previous speaker, here's an rest activity profile of a normally sighted individual. This person has no eyes, they're anophthalmic. And we see this beautiful, well, rather tragic for the individual, drifting of the rest activity cycle through time. So is there anything that can be done to people with either radical ocular damage or no eyes at all? Melatonin, of course, has been used, and melatonin agonists have been used with some level of success. Can we do something better, I guess, is the question I'm asking. Can we use the knowledge of how light interacts with the circadian clockwork to help these individuals? And I need to have a declaration of interests. I'm part of a spin-out called Circadian Therapeutics, and venture capital has funded some of the experiments that I'm going to talk about now. So basically, using an adenosine antagonist, we get a very effective clock modulation. So here's the vehicle. The vehicle was introduced here at about circadian time 6. Here's the drug, the adenosine antagonist, and you get a beautiful phase advance. And so this is the final bit of data I want to show you, which is a comparison of this adenosine antagonist with talsimeltion. Now, talsimeltion is a melatonin receptor agonist. And so we decided to compare them back to back. And in fact, it's the only FDA-approved drug for circadian regulation. And here we see an increase in the phase shift on this axis. Talsimeltion produces a shift in our mice, but it wasn't significant. The adenosine antagonist produced a nice shift, which was highly significant. So in a sense, I think this pathway seems to be a more effective target than the melatonin pathway, as far as we can tell, but it's still early stages. The exciting point, I think, is that we're now collaborating with the Blind Veterans UK, and these will be our first clinical population to test this adenosine antagonist. And in fact, we must talk about using some of your approaches on this population, which I think will be really brilliant, because there's a large number of individuals with no eyes and lots of different sort of states of retinal damage, so we should talk about that. Okay, so what I'd hope to give you some sense of in this presentation is the light regulation of PER1 and PER2. Finally, how the molecular clockwork is aligned to the external world via both Cre and AP1 activation, and the discovery of this really important element, SIC1, which acts as this brake upon the clock. Remove SIC1, and in a sense, you can shift the circadian system backwards and forwards via light. It seems as you like. I think the really interesting thing for us has been the role of adenosine. Adenosine encodes partly one's sleep-wake history, and what's very clear is that the adenosine pathway is effectively inhibiting the light activation of PER1 and PER2. So sleep-wake history and light can interact to fine-tune sleep-wake timing in ways that we hadn't really appreciated before. And then finally, I think these pathways, and particularly the adenosine pathway, and maybe also the SIC1 pathway, provide opportunities for maybe new therapeutics to try and stabilize sleep-wake timing, not only in the blind, but other clinical populations. Thank you for your attention.
Thank you, Russell, and for sharing some unpublished data. Questions, Bill?
Russell, so there was this forward mouse screen where they had the SIC3 kinase.
Yes,
and,
yes, that's also.
Can you integrate that into your...
Not at this stage, no. No, what's interesting is that, of course, if you knock out SIC3 or SIC2, it's lethal. Whereas I think what they had is sort of a slight modulation, but SIC1 mutants it's not lethal. It's very interesting because SIC1 is the only the only induced gene, whereas the other two are constitutively activated. So that's, I mean, there's going to be some interaction pathway. I can't integrate at the moment the other 6 into this system.
Hi, Russell. You have shown us a very elegant and elaborate model that is a one-cell model for what you are seeing. But you know, A1 receptors are highly expressed in astrocyte, and there is previous data that support that astrocytic coverage on the right recipient cells of the SCN changes, and that could affect the gate. So have you considered looking at that?
Well, in fact, I sort of scanned over masses of data. The in vitro stuff, of course, is done in cellular assays and a whole range of different cell types, U2S cells, for example. So it looks as though the basic biology that we're seeing in SCN cells operates in many other cell types as well, many other neuronal types, and wouldn't surprise me if it was within the glia populations as well. And I think the real power of the SIC1 mutant mouse is going to be trying to dissect, you know, all of these various pathways, what's happening not just at the level of the SCN, but at a range of other cell types. And so that's what Lewis Taylor is now working on. Yeah.
Yeah, I've got lots of questions. I'm going to go right to the back because the front people have had a bit of a domination. Here's
I have a question for your jet lag protocols.
Yes.
Did you try, instead of advancing, did you
Delay.
try to delay it?
Yes, we have done delays and it works just as well. It gets a bit more complicated with delaying because you hit different points of the phase response curve. But yes, it works both for advancing and delaying. Yeah.
Any other questions? I saw some hands. Here we go. Jason.
Hi, Russell. Can you integrate your SIK1 kinase result with the vasorepressin mutants that also have these rapid phase responses?
Yeah. I mean, I can't at the moment, no. I mean, I think there will be a series of different overlapping mechanisms, of course, and we've unmasked one. But I offhand, I can't integrate that with the vasopressin data. But you're quite right. I mean, there's a very interesting phenotype there as well. Yeah.
Any other questions? Oh, one last one. Yes, I did see your hand, sorry. Last question.
So, two very quick questions. But basically, how do you dissociate the effects of the adenosine agonist or antagonist on body temperature versus the effect
An
on
activity,
night shifting?
yeah. So could we be inducing a phase shift as a result of, for example, increasing activity, which would be one thing, and therefore body temperature? Now, we've addressed this, and I'm just trying to think about how we've dealt with it. I can't remember. It's a really good question. I'm really embarrassed. I can't remember what the findings are, but we have addressed it in ways I can't remember. And it's a shame that Artie isn't here because she would explain it perfectly to you.
And the second quick question was just to ask what type of antagonists and agonists you're actually using.
Well, it's a really good question. These are, and that's why it's, I think, quite an interesting approach, because what we did and what Sri did originally was essentially use an in vitro cellular assay with per-luck reporters and screen essentially all of the FDA-approved drugs to see if they had the capacity to shift the clock. And what surfaced was a whole bunch of FDA-approved agonists and antagonists to adenosine. So all of them have gone to stage 2, stage 3 clinical trials, and they've been developed for things like Parkinson's, and they haven't turned out to work very effectively. So what we've got are a bunch of either off-patent or slightly on-patent drugs which have gone through clinical trials that work. And so the reason we can move from mice to humans so quickly is that these have already been FDA approved in another domain, not for circadian phase shifting. And I think it represents something really quite interesting. I should say that 5 different classes of drugs across the 6,500 had an impact upon shifting the clock. And we've pursued the adenosine. There are 4 other potential compounds that could be useful. But the key point is that they are already drugs in existence, and they're either off-patent and have been clinically validated to cause no harm, but to do no good for what they were developed for. Thanks.
Thank you. And I'm going to completely abuse my position as chair to ask you to save the date for our conference next year in Lyon. This is the European Biological Rhythm Society. And on that note, I'd like you to all join me in thanking our speakers for this morning. And